Okay, welcome back to AUD 518, also known as hearing conservation. In this recording, we're going to finish up our overview of the anatomy and physiology of the human ear. We've already discussed the outer and middle ear portions, so now we will wrap it up by discussing the inner ear. All right, so the objective for this recording is for students to be able to explain in simple terms the major parts of the ear and their function. So we've already discussed the outer ear, which includes the pinna and the external auditory canal, and which ends at the tympanic membrane or the eardrum. We've also discussed the middle ear, which includes the malleus, incus, and the stapes. And now we're going to talk about the inner ear, which includes the cochlea, the vestibule, and semicircular canals, as well as the vestibulocochlear nerve. The inner ear is responsible for translating mechanical vibrations to neural signals. The inner ear can be divided into three parts: the cochlea, the semicircular canals, and the vestibule. The cochlea is housed in the anterior portion of the inner ear, and it contains the sensory organ for hearing. The semicircular canals are housed in the posterior portion, and they contain the sensory organs for balance. And the vestibule connects the cochlea and the semicircular canals, and it also contains additional sensory organs for balance. Inner ear structures are housed within the bony labyrinth, a series of interconnected spaces in the temporal bone. Inside the bony labyrinth is the membranous labyrinth that separates the spaces into two separate fluid-filled compartments. Perilymph fills the outer bony compartment and is depicted here in orange, and it's similar to fluid found outside of cells and is high in sodium. Endolymph fills the inner membranous compartment, and it is depicted here in blue, and it is similar to fluid found inside of the cells, and it's high in potassium. The hearing and balance portions of the membranous labyrinth are connected by a tiny duct called the ductus ruinus. Thus, the membranous labyrinth forms one continuous fluid system filled with endolymph. If we unroll the cochlea, we can see that the membranous labyrinth separates the cochlear duct into three compartments. The scala vestibuli is the upper compartment filled with perilymph, and it contains the oval window where the stapes footplate is connected. In the middle, we have the scala media, which is the middle compartment, which is filled with endolymph. At the bottom here, we have the scala tympani, which is the lower compartment filled with perilymph, and it contains the round window. The scala tympani and the scala vestibuli communicate through a small opening called the helictrema at the apical end of the cochlea. The cochlea is a snail-shaped structure built around a bony core called the modiolus. The cochlea is 5 millimeters high. It is 9 millimeters wide at the base and smaller at the apex. The cochlea contains 2 3/4 turns. The unrolled cochlear duct is 35 millimeters long. Okay, so here we've got an image that's showing us the modiolus. We've also got in this image the osseous spiral lamina, which is the shelf of bone that extends outward from the modiolus and supports the medial edge of the basilar membrane. We can also see the basilar membrane depicted here, which is the fibrous membrane that runs horizontally from the osseous spiral lamina to the spiral ligament of the outer wall of the cochlea. Okay, it's also important to note that the basilar membrane is narrower and stiffer at the base and wider and less stiff at the apex. The osseous spiral lamina is made up of two plates of bone with a space between them, and the auditory nerve fibers travel through this space to contact the sensory hair cells. The central processes of the auditory nerve fibers come together to form the eighth cranial nerve, which exits from the cochlea through the internal auditory meatus. So we have here depicted the spiral ligament, the fibrous connective tissue on the lateral wall of the cochlear duct that supports the lateral edge of the basilar membrane. We also have Reisner's membrane, which is a thin membrane that separates the scala media from the scala vestibuli. We also have the stria vascularis right here, which is which generates a positive electrical voltage in the scala media. We also have the organ of Corti depicted here, which is the complex structure comprised of the sensory hair cells and other supporting cells. It sits on the basilar membrane. We've got the tectoral membrane, the gelatinous structure that overlays the cells of the organ of Corti, the spiral ganglion, which are the cell bodies of the auditory nerve fibers that innervate the sensory hair cells. Again, these are the cochlear structures. Spiral ligament, Reisner's membrane, stria vascularis, organ of Corti, tectoral membrane, spiral ganglion. Okay, the organ of Corti is a complex structure comprised of the sensory hair cells and other supporting cells. So the organ of Corti consists of one row of inner hair cells, shown here, one row of inner hair cells, and three rows of outer hair cells, shown here, 1, 2, 3. Okay, here we've got our tectoral membrane on top, and then we've got our reticular lamina. All right, so the inner hair cells, we're talking about one row along the length of the cochlea, as just shown, which consists of about 3,500 inner hair cells, and these are flask-shaped. The cell bodies contain mitochondria and other structures that are important for the transduction of fluid vibrations into neural signals. In addition to the inner hair cells, we have 3 rows of outer hair cells along the length of the cochlea. There are 12,000 of these babies, and these are cylindrical in shape, and the cell bodies contain structural proteins that allow the cell to change length in response to basilar membrane vibrations. Okay. Stereocilia at the tops of the inner hair cells and outer hair cells are important to their functions. The outer hair cell stereocilia are embedded in the tectoral membrane, while the inner hair cell stereocilia are not. And you can see that here. So there's 3 rows of our outer hair cells, and you can see here that they will become embedded in the tectoral membrane when they make contact, but the same is not true for the inner hair cells. Right? So you'll recall that the basilar membrane is stiffest at the base and least stiff at the apex. Due to this stiffness gradient, high frequencies cause vibrations near the base and low frequencies cause vibrations near the apex. Thus, the cochlea performs a tonotopic mapping of frequency to place along the basilar membrane. So the vibration of the stapes foot plate sets up a pressure wave in the cochlea. This results in a wave of basilar membrane displacement that travels down the cochlea. We call this a traveling wave. The wave peaks in amplitude at the point along the basilar membrane with ideal stiffness for that frequency. The wave slows down as it approaches the peak, and any leftover energy in the wave is dissipated or damped. We have our base and we have our apex, and this is showing us the traveling wave pattern on the basilar membrane at one instant in time. So this is where the peak of the traveling wave is. You can also see that in this demonstration here. So you can see you've got the tympanic membrane, the malleus, incus, and the stapes. The stapes generates the pressure wave, and the peak of the wave occurs somewhere in the middle of our unrolled cochlea here, and it dampens or dissipates. Okay, the location of peak displacement depends on the frequency. Lower frequencies peak toward the apex, and higher frequencies peak toward the base. So the tympanic membrane is set into vibration by sounds. The stapes footplate moves in and out of the oval window, and the basilar membrane moves up and down. So how are the hair cells activated then is the next question. So when the basilar membrane moves upward, a shear force is generated between the reticular lamina and the tectoral membrane. This force causes the stereocilia to bend in the lateral direction toward the outer wall of the cochlea. This lateral bending opens specialized pores in the stereocilia and activates the hair cells. When the basilar membrane moves in the downward direction, the stereocilia bend in the medial direction toward the medialis. The medial bending closes the specialized pores in the stereocilia and inhibits the hair cells. So activation of the inner hair cell causes a chemical reaction in the cell. This reaction causes a special substance to be released at the base of the inner hair cell. The neurotransmitter then activates the auditory neuron. Thus, the inner hair cell transduces mechanical vibrations into neural signals. The activation of an outer hair cell causes it to change in length by stretching the structural proteins in the cell body. During the excitatory phase, the cell shortens, making it easier for the basilar membrane to overcome frictional forces that resist its upward movement. During the opposite phase, the cell elongates, exerting an extra force on the basilar membrane. Thus, the outer hair cells amplify the vibration of the basilar membrane and serve to improve hearing sensitivity. The motor activity of the outer hair cells is known as the cochlear amplifier or active process in the cochlea. The most common type of permanent hearing loss occurs when there is damage to outer hair cells and the cochlear amplifier is lost. So innervation refers to the distribution of nerve fibers to an organ or region of the body. Afferent innervation refers to nerve fibers that send signals upward from the cochlea to higher auditory centers. And efferent innervation refers to nerve fibers that send signals from higher auditory centers downward to the cochlea. There are approximately 30,000 afferent auditory nerve fibers in humans, and they are divided into two types. There are type 1 fibers, which carry information about the sound stimulus. Type 2 fibers have many connections to outer hair cells, and each type 2 fiber connects to 10 to 20 outer hair cells. Each outer hair cell is contacted by about 10 type 2 fibers. The function of these type 2 fibers is unknown. Here we have a depiction of the type 2 fibers that connect to many, many outer hair cells. You can see that here. There are approximately 1,600 efferent auditory nerve fibers in humans. They are part of the olivocochlear bundle that descends from the superior olivary complex to the cochlea. They have some contact with outer hair cells directly. Others contact afferent neurons, and they are thought to modulate cochlear activity. So we've talked about the tympanic membrane set into vibration by sound. We've discussed the stapes footplate, which moves in and out of the oval window, creating a pressure wave or a traveling wave. We've talked about the basilar membrane that moves up and down, the outer hair cells, which amplify the basilar membrane displacement, inner hair cells, which transduce basilar membrane vibration to neural signals in type 1 fibers. And next, we're going to wrap up with the auditory nerve and how it sends coded information to the brain. Auditory nerve fibers from all parts of the cochlea come together in the medialis to form the cochlear portion of the 8th cranial nerve. Once the sound travels from the outer, middle, and inner ear, the next step is the brain. There are 3 major parts, including the brainstem, cerebellum, and cerebrum, or the cortex. The brainstem is a central core of the brain. It controls involuntary processes like heart rate and breathing. The brainstem contains nuclei of 12 cranial nerves, and it routes information to the cortex. Components of the brainstem include the medulla oblongata, the pons, the midbrain, and the thalamus. In addition to the brainstem, we have the cerebellum, which means little brain or small cabbage. It is responsible for coordinated movement. We also have the cerebrum, or the cortex, which is where the high level of processing of information occurs, motor, planning, and control. And the cerebrum, or the cortex, also mediates thought, language, emotion, memory, and learning. Components of the cerebrum, or the cortex, include the outer cerebral lobes, the inner white matter, deep nuclei, and limbic structures. Okay. Lobes of the cerebral cortex include the frontal lobe, the parietal lobe, temporal lobe, which is what we are primarily concerned about, the occipital lobe, and the limbic lobe. Sound travels from the outer to the middle to the inner ear, then continues to travel along the vestibulocochlear nerve to the cochlear nucleus, the superior olivary complex, the inferior colliculus, the medial geniculate body, and finally ending at the auditory cortex.